and bioequivalent studies. Although we are very familiar with this, let us have some deep industrial approach towards this bioavailability and bioequivalence by Srinivas Reddy Garu. Let us have a brief introduction about him. So, Srinivas Reddy Garu currently working as a manager in a quality control department in Aurobindo Pharma Limited. He also worked as a group leader in the quality assurance department in Apotex Research Private Limited, and also worked as a research associate in quality assurance department in Trident Life Sciences, and as a research analyst in pharmacokinetic and statistics in Bioserve Clinical Research Private Limited, and as a project trainee in analytical R&D Dr. Reddy's Laboratories. He has got his MPharm in December 2004, and he also attended many workshops and training programs, and few of them like reducing human errors, human reliability training program, global audit certification program, drug information association, which is an 8th annual conference, and many workshops on good laboratory practice. He also acted as a trainer conducted by ADICO India. He participated in the National Seminar in Pharmacovigilance in India organized by Indian Pharmaceutical Association. He also participated in internal audit training ISO 9001-2000 conducted by Biru Vertes, Hyderabad. He also conducted many internal audits, right? And he also working as a challenging person currently in Aurobindo Pharma Limited. So this is a brief introduction about our speaker, today's guest speaker, Srinivas Edruri Garu. And let us start this informative and uplifting session on bioavailability and bioequivalent studies.
Thank you, Sargama.
Thank you, ma'am.
Now I would like to welcome our speaker, Srinivas Reddy to for your presentations.
Okay, thank you, madam. Thank you for your welcome.
Thank you.
So, as you said, bioequivalent studies is not the new topic. So, but most probably, many people may are not aware how to conduct bioequivalent studies and what will happen during bioequivalent studies conducting. So, in our academic, we don't have these facilities or provision to conduct the bioequivalent studies. So we can have the laboratory facilities, HPLC or whatever it may be, but we don't have the facilities for human studies. Okay. Yes. So just I will go through all the, I will explain briefly, and because it is a big topic. So from starting to and ending calculation, I will explain the how bioequivalent studies or bioavailability studies conducted. So sure. Bioequivalent studies and bioavailability studies are the same, but calculation is different. That's all. Okay. Okay. So then just if you have any doubts at the end of the session, you can ask free to ask the questions. Okay.
Sure, sir.
Dear participants, you have a session. After the end of the session, you'll be asked some questions.
Just note it down the slides and wherever you have doubt and you can ask. Okay.
You please give in the chat box.
Okay.
Okay, sir.
Okay. So anyhow, already introduction is I am working as manager in quality control laboratory. Previously, I worked in bioequivalence department, Apotex, and as well as Bioserve clinical research. Okay. Sure. So the main objective of this seminar or presentation is so purpose of the bioequivalence studies. What are the bioavailability? Even the bioequivalence it is written. So both bioavailability and bioequivalence we can. So then purpose phases of the bioequivalence studies. So whatever it may be the studies, we have some phases. So that's why I will explain about what are the phases of that studies. Okay. Yes. So then the main contents of this presentation is so drug discovery, brief drug discovery and development. What are the generic drugs? So phases of bioequivalence studies. So phase of purpose of bioequivalence studies and phases of bioequivalence studies. Okay, so these are the three types of whatever you were the studies, whatever either clinical studies or clinical trials or whatever it may be, it will have three phases. So one is clinical phase, conducting the study, and analysis part and calculation part. Okay, these are the three parts we have the any clinical studies. So then why we do this? Why these studies? For regulatory submissions. So that means for approval of the drugs. This is a part of one module in the submissions. So it's a mandatory module for the submission. So then we will just brief introduction of the discovery that you know very well about the drug discovery. And so life cycle of drug discovery have the 4 phases. Okay. So discovery and research, development of the molecule, so regulatory review and approval, then commercialized in the marketing. So that it has to go to the market. These are the 4 phases of the drug development. So generally, you know, just nowadays it is decreased, but generally from so core molecule to clinical phase and into the market, it will take around 10 years. So nowadays, because of sophisticated techniques, it may reduce some years, but general term is 10 to 12 years generally. Based on the sophisticated techniques, we may reduce the time period also. Okay. So then this slide, just if you observe that one, so suppose chemical entities, we may develop thousands of molecules. In that, it will go to either 10 to 20 molecules to the clinical phase. Then finally, it will go to the 1 molecule to the market. So we have so much work behind a drug coming to the market. So these are the brief 4 pages. Discovery research, you have the identify the suitable target. Suppose what is our discovery research? Where we have to target the molecule. So then it will depends on the how target. This is the just discovery phase. And again, drug development. So once you discover the target, then you have to go for the development of the molecules. So then you want to develop the development of the molecule, you're testing and optimization of the selected compounds to identify the lead structure. So then structure, what is the structure and what is the, so how it will so go and work into the humans. So those drug development is one of the phases. And again, suppose even it is not enough to development of the drug. So what is our main aim? It will reach to the patients. Okay, then if you are it reach to the patients means you have to take the approval from the regulatory. So then ours is the CDSCO is our Indian regulatory. But if you want to market to the other countries, each country having the different names. That is FDA is the US and EMEA European, like the TJ is Australia. So like that, Canada is having their own regulatory guidelines. So according to wherever you are going to the market the drug, you should follow their regulations. Okay. So then marketing. So regulatory approval is one of the process. Then you have to market the drug. Then only, so it is having the some. You will get, suppose if you want to develop the drug, if you won't market the drug into the markets, then you will not get any single money. So it's totally waste of the time and money. So that's why our target is discover is good molecule and it will reach to the patients with good. So we'll earn the money with good incomes. So everybody industry will, otherwise nobody will go for industry. So even nobody will start the industry. Our aim is, so you have to develop the drug and you have to market, we'll earn the money with good drugs which are helpful to the patients. So these are more the main motto of the industries. So especially this will comes under the research and development. Okay. So then come to the main topic. Generally, before going to the biochemistries, so we have the generally we have to bifurcate the clinical trials and bioclinical studies. Okay. So these are we generally have the 4 phases of the clinical trials. So generally, we do clinical trials for new molecules. So keep in mind new molecule and generic drugs. So generally, bioclinical studies we go for generally generic drugs. So generally, clinical phase are 1, 2, 3, 4. So 4 is the first post marketing. So 1, 2, 3 we have to conduct for new drugs, that is branded drugs. So before going to new molecule into the market, so you have to cross these 4 phases, 3 phases studies. Then you have to market the drug. After marketing also you have to conduct the phase 4 studies for the efficacy of the molecule. So now coming to the just general introduction of the what is the branded drugs and what are the generic drugs. So these are the qualities of the branded drugs, also called as innovative drugs, that is new molecules. So initially marketed as a new chemical entities supplied by one drug company because they will take the patent and they will supply only So that company only supply the drug. So nobody is having the rights to prepare that molecule. So they will take the patent, and some years based on the molecule, they will take some years of the patent. Then they will only have rights to sell the drug, that branded drug, into the market. So because so they have spent lot of money in that drug discovery. So then sold under company's trademark. So cost is very more, because when a new molecule come into the market, so it will have it will have more cost, because of so more money they spend on the research, and when research to come into the market. So that is until come into the market, they will not get any single penny for that particular research. So once it come to the market only, they will get the money from the market. So that's why cost will be more when branded drugs. So once patent is completed, so any company can prepare that molecule. So then they can approve, they can take approval from the respective regulatory, and they will market the drug. So then, so generic drugs will come into the picture. So generic drugs is generally almost a copy of the branded name drugs. Suppose if you take omeprazole, so different brand drugs are there. Omeprazole, you have the many generic drugs. Okay, so it's produced after original patent expires, maybe supplied more than one company. So maybe sold under active ingredient names, cost is less. Why? Because no need to spend the discovery cost and as well as a clinical trial cost. So okay, that's why. So they will develop very cost is very less for developing the generic drugs. So then, what is the requirement of the generic drugs? So same active ingredient. That means paracetamol means the generic drug should be paracetamol. So same route of administration, that solid dosage forms or oral dosage form, that solid or that is route of administration means either oral or intramuscular. Then IV, intravenous, we don't develop any generic drugs, because intravenous is having the 100% bioavailability. Directly we will go for the systemic circulation. Then we don't develop any generally. So bioequivalence we don't conduct for the injections. So either suspensions, tablets, capsules, we develop the we conduct the bioequivalence studies. So same, that means same dosage forms. Either the innovator is the tablet, we have to develop the tablet. So then same strength, 10 mg means 10 mg, 20 mg means 10 mg. So same conditions of use. So that means condition means it has to take fasting or with meal or without meal. So these are the same conditions. So whatever the conditions apply to the innovator molecule, same should be applied to the same type you have to develop the generic molecule also. So compared to the brand, so what are these qualities should be compared to the branded drug. So then come to the exactly, I will explain this slide. What are the generic difference between the generic and branded drugs? What? So then if you generally, you know, this is are the terms of FDA terms. So NDA means new drug applications, and ANDA means abbreviated new drug applications. Generally, abbreviated new drug applications are the generic drugs. So new drug applications here are the new molecules. That means innovative drugs. So then the chemistry of the molecule is same. So then manufacturing process is same, and controls also same as that of both innovator and generic drugs. And labeling, labeling means, you know, the labeling will come for the any drug, you should have some labeling. That means, so what are the pharmacology actions, what are the directions of indication of use, and how to take the medicine, what is the storage conditions, everything will come under the label. So both the generic and innovator drugs should be same labeling. So testing, that means testing means testing of the drug for their quantitative analysis or qualitative analysis, or assay or impurities as present or not. Those testing is common for both drugs. So then so come to the, if you come to the innovator drugs, so they should do animal studies, clinical studies, that phase of studies, and bioavailability. So when you come to the generic drugs, so these animal studies, clinical studies may not required. So only if you conduct the bioequivalence studies enough to submit the application to the regulatories. So that's why, so the cost automatically decreased, it come to the generic drugs. So this is the basic difference between the new drug and generic drug, and where bioequivalence will apply for generic drugs. So then come to our topic. So bioavailability. So as you know, everybody will know the definition of the bioavailability. Same definition I have given here. There is no new definition. So bioavailability refers to the relative amount of the drug from an administered dosage form which enters into the systemic circulation. So bioavailability we see generally oral dosage forms. Okay. So when it reaches systemic circulation and rate at which the drug appears in the systemic circulation, that means what is the availability of the drug in the systemic circulation. So that is called the bioavailability. So just the difference between the bioavailability bioequivalence is, so whenever you come to the bioequivalence, you should have the compare with innovator product. So then only we can see the both are equivalent or not. So slightly difference between the bioavailability and bioequivalence is definition. So the definition is the absence of significant difference in rate and extent to which the active ingredient or active moiety in pharmaceutical equivalents or pharmaceutical alternatives becomes available at the site at drug when administered same molar dose under similar conditions. So this is very important. Same molar dose and similar conditions. So that means what drug is 10 mg. If you give the generic drug is 20 mg, we should not compare because it is not the similar doses. So under similar conditions, what are the conditions applicable for innovator drug? So it should be applicable to the generic drug also. Then only we can compare both the drugs. Then we can say both are bioequivalent or bioinequivalent. So, okay. So then come to so this is generally just a pictorial diagram of comparison. If you take the red is the one innovator and blue color is the generic drug. So when you compare that, both should be equivalent. Then only we can say so both drugs are equivalent. So then different phases of the bioequivalent studies. So clinical services from final, where do you when do you do the bioequivalent studies? Okay. So what is the purpose of bioequivalent studies? So generally clinical service form to the final market form. So generally suppose already completed formulation, when we are going to the final market form, we can conduct the bioequivalent studies. Change of the formulation. Then suppose my current dosage form is capsule. When I change to the tablet, so then we can conduct the bioequivalent studies, and if you prove the bioequivalent, then we can go. వస్తున్నా అది తీసుకురా చార్జర్.
ఏ చార్జరు దీంట్రా?
Is there any power cut issue, sir?
Power problem. Sorry for trouble. Sorry for the inconvenience.
Sir, parallel, sir.
Yeah. Thank you. So the purpose of bioequivalent studies, when we do the bioequivalent studies, okay. So when we clinical form to the final market form, so change of the formulations from tablet to capsules, then generic formulations. As I said, generic formulations, most of the industries, So do bioequivalent studies for generic formulations. Okay. So most of the 90% of the industry, they do the bioequivalent studies for generic market. So different phases of the bioequivalent studies. So generally we have the 3 phases. So any bioequivalent study or clinical study has having the 3 phases. That is, so clinical phase and bioanalytical phase and pharmacokinetics and statistics, which is the calculation part. So then clinical phase, we have the protocol preparation to the sample transfer to the bioanalytical lab, that is testing laboratory. So these phase we will call as the clinical phase. Then after that, whatever the molecular analytical moiety, so you have to estimate from the collected sample. So those will be the bioanalytical phase. Okay. So then after bioanalytical phase, concentrations will come. Then we have to prove the bioequivalence. So you have to calculate some. We have to calculate the concentration with some parameters. So based on the parameters comparison, we can say that is it bioequivalent or not. So these are the 3 phases of the bioequivalent study. We'll go to the one phase to phase. So before that, going to the that phases. So if you want to conduct the bioequivalent studies, you should have the approval from the DCGI. In India, if you have to conduct the bioequivalent studies, you should have the permission from the Drug Control General of India. So this is the our health department, which is in Delhi. So even you should have the permission to conduct the study. You should have the some facility. So to facilitate conduct the bioequivalent studies, like your lab. So like like your lab is the conducting the general laboratory purpose. So like that, you should have the some facility to conduct the bioequivalent studies that should be approved by the DCGI prior to conducting the studies. If you want to start off any study, that facility should be approved by the DCGI. That means you should get permission. Even bioanalytical laboratories also, you should get permission to conduct the testing for biosamples. So then BENOs, that means bioavailability no objection certificate for export purpose. So then if you want generally bioequivalent generic drugs, many companies, they don't market in India. So because they don't get, they will get less money when they market into the India. That's why many companies, they sell the generic drugs in so US or Europe countries or okay. So generally 5 types of BNOs export licenses are there. So then new molecules. So but new molecules means already available in out of the countries, which is not available in our countries. So these categories is one category. So another category is new drugs approved in India with a period of 1 year. That means the drug is already approved in India, but only 1 year the period it is in the Indian market. So then come to the new drugs approved with period more than 1 year and less than 4 years. So because the drug is approved 1 year over and less than 4 years. So next is drug product is modified release from the respect of their approval status. So one more is the T license BI based is older drugs, which are already available in the market longer period. So that license is different. So these license, 5 types of license will have the, some are the documents is common. So some are documents is not required. Suppose if you take the new molecules, so then approvals are approval type of the documents submitted for approvals are the different. Approval process is same, but type of document submission is the different. Okay, so the keep in mind. So approval is mandatory from the DCGA. That means CDSCO. So in India, if you want to conduct the bioequivalent studies, so before conducting, you should take the permission for that particular study. Then only you have to conduct the bioequivalent studies. These are the 5 types of licenses. Licenses means approvals, so which are provided by the DCGA. Then study designs. So if you want to conduct the studies, you should have some study designs. Single dose, 2-way crossover, fasted. So then fed studies, then all
Sir, we couldn't hear you, sir.
So now is it audible?
Yes, yes, sir. It is audible now.
Okay, thank you. So single dose, these are the different types of studies, that means designs. Suppose if you want to conduct, you should have some design. So these are the some designs generally we follow to conduct the bioequivalent studies. So general common general bioequivalent general designs I am explaining here. There's so many more designs which are complex designs, but generally, so 2x2 crossover design. So then in the generally most of the 90% of bioequivalent studies will conduct this 2x2 crossover. Okay, in that you having the two formulations, that means test and reference we will test. So 2-period study. Okay, 2 sequence means either RT or TR, or then 2-period you have to give. That means if you give the test drug in 1 period, you have to give the reference drug in another period. These are the 2 sequence and 4-period design. So that means you have to conduct the, then generally we will go for here TRRR. That means 2 reference molecules and 2 test molecules. So if you want to conduct the study at a time with the 2 molecules, 2 different, if you want to compare the 2 different test products with 2 different reference products, then we will select this 2 sequence and 4-period design. So then 4 sequence and 2-period design. Okay. So this period, this 2 periods only will, this is also same type of study. So because we will compare the 2 test and 2 reference products, but the number of periods is very less. So that means the 2 periods only we can complete the study. So then 4 sequence and 4 period. So, okay. So this is also same type of, we'll compare the 2 test and 2 reference, but with different sequences. So simply minded that sequence is the randomization of the drugs before going to give the subjects. Then TTRR means, suppose I will explain in earlier the coming studies. So what is the period and what is the that is period means. So then this will comes under period clinical phase only. So then if you want to conduct generally in our laboratory also, any experiment you have to conduct. Okay, so you should have some procedure. Okay, so that same is applicable here bioequivalence also. So if you just think that our dissolution studies, in our dissolution studies we will conduct in laboratory, we should have some procedure. What are the time points, what are the media, and what equipment you have to use. And here generally just equipment is our human body. So like just you can compare the dissolution with our studies, that here we are conducting like type of dissolution studies in vivo. So the we will call dissolution studies or in vitro studies. Then here we will conducting the same pattern in vivo. So in humans. So our instrument is the human. It's like that our procedure, and but so in laboratory, so we can conduct the bioequivalence. That means in laboratory we can conduct laboratory experiment like dissolution with some procedure. So like that, here also we should have some procedure, but we cannot conduct as that of the laboratory. But it's ethical because we are doing the bioequivalent studies in humans. That means some population, that is humans we are doing. That's why you should have some procedure for pre-approval procedures, and that pre-approval process should take before conducting the bioequivalent studies. So these are the, yeah. So one by one we will discuss. So protocol preparation, that protocol is like our experimental procedure. We'll have the all details in the protocol. So then ethics committee approval. So it is one, this is not required for our general laboratory testing. So ethics committee means ethics committee is one of the committee before going to take this approval from the regulator. That means CDSC or DCCA. The protocol should submit to the DCCA. Then on that, based on the our protocol conditions and our what are the time points and what are based on our protocol preparation, based on it is sufficient or not, they will check everything and they will give the committee will give the approval to the protocol only. So then after that you have to take the permission from the DCCA, then you start remaining process. Okay, before going to take approval, your protocol should be ready. So to submit to the DCCA. So based on that only, based on your experimental procedure, so they will give the approval for that particular protocol. That means particular protocol means you can do only one molecule, that is one study for that particular protocol. So like one experiment. So if you want to conduct more studies, that more bioequivalent studies, you should get individual approval for each bioequivalent study. Okay. So then informed consent process, selection of the subjects. Subjects means then humans. So test article packaging, test articles means our drugs. So drug administration process, sample collection, separation and storage, adverse event monitoring. So it is one of the important adverse event monitoring, and transfer of the samples to the bioanalytical laboratory. So if you see that these are the different steps in the clinical phase. That means protocol preparation to transfer the samples to the bioanalytical laboratory. So then come to the protocol. So you see anywhere you can search in net also, it will give some definition. So what are the definition? ICH GCP. That means good clinical practice is one of the Bible for generally clinical studies, or universal guidelines for the clinical studies. So what is the protocol means? A document that describes the objectives, design, methodology, statistical considerations, and organization of a trial. So that means, so design objectives, design methodology to the statistical calculation, it should be covered in that protocol. So if you read the protocol, you should understand that what from starting stage to ending stage, what are the acceptance limits, everything you understand when you read the protocol. So generally some bioequivalence protocol will have these contents. Okay, just if you see that one. So protocol, what is the overview? What is the pre-documentation required? That is objectives, investigators. So investigators is required. That means investigator means generally head of that particular clinical phase. Okay. So how to select the subjects, and what are how many number of the subjects to recruit, and what is the standardization and blinding design, so administration of the drugs and everything, and measurement methodology. So from that measurement methodology means bioanalytical part. So then analysis of the data means comes under the pharmacokinetic part. Just if you see that one. So from protocol overview to the up to the measurement of the prior to measurement of the methodology comes under clinical phase. So measurement of the methodology comes under bioanalytical phase. Analysis data comes under the pharmacokinetic phase. Okay. So these are the different phases what I explained earlier. So you should cover all the phases of the clinical study or bioequivalent study in the protocol. So then ethics committee. So what I said, ethics committee, why is requirements? So you should not conduct the experiments in humans as you like. So that should be some ethical procedures are there, because you should not treat humans as like animals. Okay, even if you want to conduct the animals also, you should get approval. Okay, so like that now we are testing our drugs on humans. So that's why you should get permission from the ethics committee. So these generally ethics committee, either IRB or institution review board or ethics committee, you can call whatever it may be. So previously, when 5 or 6 years back, not really 10 years back, so we don't have any ethics committee approval by the DCI. So now even ethics committee, if you want from also, that ethics committee should have the approval from the DCI. So these are the members that committee should have the approval. So then only those committees should have the eligible to review our protocols. So generally chairperson will be the ethics committee will contain both medical and non-medical people, and one lay person should be there and one lawyer should be there. So like that different multiple sectors person will be in that particular ethics committee or IRB, institutional review boards. So what is the main aim of these review boards? So main responsibility is to ensure the protection of the rights, safety, well-being of human subjects. Suppose if you submit the protocol to the ethics committee, they should approve the protocol. Then only you will have right to submit that protocol to the regulator. That means approval process for DCJ. So that means what they do, so they review the protocol, and is the protocol contents are sufficient or not to conduct the bioequivalence study. So then they will conduct one meeting. So in that meeting, that persons will be present. They will present the protocol. If everybody agree, that they will form a quorum. Quorum means limited. The different. Suppose If you have 12 members of the subjects, no need to attend all 12 members ethics committee to approve the protocol. Some quorum will be formed. That means minimum 5 members they will attend is sufficient to approve the protocol. If they agree, that means if they have any queries, they will send the mail to the organizations. So once you resolved only, they will give the approval. If it is not sufficient, that what are the contents in the protocol are not sufficient to conduct the bioequivalence study, or if any problems are there in that contents, so then they will reject the protocol. Then you have to resubmit the protocol to the ethics committee. So okay, this is the main purpose of the ethics committee. So then comes to the informed consent process. So when you are conducting the experiments, no need to take the permission from the instrument or whatever it may be. So then here you have to take the consent. That means when I am taking the humans, you should take consent from the humans, because I am voluntarily participating in the, you have to explain everything to the volunteer. When you agree only, you should recruit the subject. So that what are the informed consent involves providing the relevant information of the volunteer to, that means relevant information means, so what is my purpose of the study, why I am doing that particular bioequivalence study, what it contains, what are the adverse events if you take the drug, and what are the serious adverse events if you take the drug, and what is the, how many days you should be here, and what is your compensation we will give. Okay, so whenever voluntarily, if you agree only, you to take, you have to recruit the volunteer. Otherwise, you should not, if you reject, you should not forcibly recruit the volunteer into the study. So informed consent is communication process between the researcher and participant. That means researcher is organization, participant is the subject. So this is the between the research and participant starts before research is initiated. That means informed consent process should be present to the volunteer. You have to take the informed consent before start of the study only, because after start it should not be, you should not take the consent after inserting the drug, after administering the drug to the patient or volunteers. So one more thing I want to tell you, generally bioequivalence studies, most of the times 99% we will conducting the healthy volunteers. That means they are not the patients. Okay, so that's why, so before start of the study only, you should take the consent from the volunteers. So volunteer subject are the synonyms. So continues throughout the duration of the study. Okay. So consent must be in language that subject understands. Suppose, we should have some doubt. Suppose, can we take only Telugu people? Can we take only English people to the? So no, you can take any language people to the study. So, but you should explain the participant in the language which is understood by the volunteer. Suppose you don't understand the Telugu, you should not explain in Telugu. So what are the languages, either English or Gujarat, Punjabi or Kannada. So whatever it may be the language you are recruiting the subject or volunteer, you should explain your details to the subject in that particular language only. Okay. And also after explaining, so nowadays, previously there is no audio video recording. So now that consent process should be audio video recording, which is auditable by the regulator departments. Okay, that means each consent when you are taking the subjects, you should record that subject. What are you explaining? In these subject questions are there, you are resolved completely. So that audio video recording should be archived for audit purpose. So that after that, that informed consent form should be signed by the particular subject. Then only, so that sign is the indication of accepting the volunteer to participate in the particular subject. So this is in our clinical studies of bioclinical. This process is very, very important process. Okay. So then selection. So selection.
Sir, we cannot hear you, sir. No, sir. So once check like unmuting yourself and so muting yourself and unmute again. No, sir. Still we cannot hear you. Sir, we cannot hear you, sir. Hello? సార్ ఒకసారి మీరు మ్యూట్ చేసుకుని మళ్ళీ అన్మ్యూట్ చేసి చూడండి సార్. చెప్తున్నాను. ఇదేంటి ఇది పోవట్లేదేంటి?
Hello. Hello.
సార్ ఎస్ సార్.
Madam, it is audible?
ఎస్ సార్.
So some interruption is there. Okay.
Yeah.
So generally selection, why you have to select the subject, screen selection of the subjects. So we cannot take the subjects as suppose he is having disease, or we should not take the subjects. So as I said, you have to take the healthy volunteers. Then how do you know the subject is healthy or not, any disease is there or not? So that's why before going to the take the subject, you should, once he accept the consent, you should go for some screening process. Okay, so that means why you have to go for, so what are the subjects you are taking, we should minimize the physiological variability. That is, so internally, so each person is not having the same physiological conditions. So that's why, so as minimum you should decrease the variability of the subjects by testing some parameters in the before going to conduct the study. So those based on those parameters is fine, then only we will take the subjects into the study. So then we should have some inclusion exclusion criteria. That means we have some checklist. Suppose if you meet that particular criteria only, you should take the subject into the study. So for that, you have to go for some general screening. So that general examination will contain the medical history and physical examination, or ECG, chest X-ray you will take. And after that, you have to conduct the blood test. That means CVP, that means blood tests and everything you will collect. Some tests we should not go for all CT scans, everything. So that you should conduct the minimum tests to check the person is healthy or not. So that minimum tests are, he is passed the, that means he is test results are okay, then you will enroll that particular subject. So then when selection, generally bioclinics will be conducted in 18 years or older, and generally will take up to 45 years. So then the BMI should be 18.5 to 30 kgs per meter. That means, so that what are the general requirement is this one. So maybe based on the different studies, some excursions will be there. So, but this definitely bioclinic study means we will go for 18 years to 45 years. So then gender either males or females. So generally female studies are very less, just any hormonal studies, it is specific to the females. So then only we will recruit the females. Otherwise, most of the bioclinic studies will conduct on males. So smoking status also you have to check the smokers or non-smokers. So the number of subjects to be participated. So this will be mainly depends on the our variability of the drug. So suppose if variability is more in physiologically, that means suppose most of the drugs, some drugs may having a more variability in our person to person. So in that case, you have to go for more number of subjects. So because otherwise that study will not be a pass. That's why. So that will be how the sample size also you have to calculate based on the drugs. So then come to the administration. So once consent is completed, screening is completed, we enrolled the subject. So, okay. So once we enrolled the subject, so how do you check? How do you check the our test drug and difference reference drug the same? So then you have to administer drug for that particular subject. So like that in dissolution, we drop the tablets into the bowel. So like that, you have to drop the not I cannot say drop, but you have to administer the drug to the particular volunteer. So that means once you administer the drug, so automatically it will go to the body. Then that what are the physiological variation that dissolve the drug, it enters the systemic circulation. Then after that, you have to collect the either blood or urine or serum. So then that analysis will be further process. So that's why if you want to collect the if you want to go to the process, then you have to administer the drug first. Like that, what I said, dropping the tablet into the dissolution. So generally, before when you want to administer the drug, you should take some water. Like that. So how much ml to take is depends on the regulatory market. Suppose if you want to conduct the study for US, so generally it is 8 ounces. That is 240 ml with 240 ml of water. So you have to take the medicine. So each volunteer equal amount of water you should give for at the time of administration. Okay. So like that, EMEA they will prefer 150 ml. So then suppose some countries, so they don't take the tablet with water. Sometimes we will conduct the apple sauce studies. Generally, these studies for omeprazole drugs, we will do the apple sauce studies. What they do, what are the pellets they will put into the apple sauce and they will take that medicines. So for that purpose, if any specific is there, then we will conduct the other fluid. So generally, we will administer the drug with water only, most of the times. So then once you administer the drug, what will happen? It will go into the body. So then you have to collect the either blood or urine. So generally, most of the 99% will go for blood only. So urine studies, especially if any elimination is more, so in urine, so then we will go for urine drugs. Generally, we will go for blood only. So then generally, so then we will collect the blood. So like that, if you take the, for example, I can give the dissolution profile. So how can we go for dissolution profile? So then either half an hour, 1 hour, 2 hours, 3 hours, like that, we will collect the sample, we will go for profile. So same can we apply here, we can apply here, because so when we administer the drug, then you have to collect the samples. So the sample collection will be specified in the protocol. Either 0 hour, anyhow, you will collect before administer the drug, you will take the 0 hour. That means in that there is no drug. 0.5, 1, 1.3, this is depends on the Tmax. So that means the drug is early, that's it eliminates early and it will reach maximum concentration early. Then we will shorten the interval between the two points. Suppose it is more half-life is there, and then we will go for more time interval between the 2 points. So generally sample collection at specified time points we will collect. So then, so after collecting the sample, what you have to do? So our bioanalytical method is directly estimation from the blood. So then no need to worry. Same blood will transfer to the bioanalytical laboratory. Suppose if it is developed in serum, then you have to separate the serum from the blood. How do you separate? If you centrifuge the blood, that it will come the serum or plasma. So if our method is plasma method, then you have to separate the plasma from the blood. Then you have to send that plasma to the bioanalytical laboratory for testing. So the next process is centrifugation or separation of the plasma or serum from the blood. So then storage of the sample. So generally biosamples will be stored under -degree centigrade. Why? Because otherwise the drug will be degraded. That's why most of the times, so we'll store at either -30 degrees or -80 degrees. So, okay, so that's otherwise the drug will be degraded, that will be useless. So for analysis. So then one of the most important thing in the bioequivalent studies or any human studies is the adverse events. Suppose if you take any drug, any allopathy, so it will have some the side effects. Suppose for a best example is mannitol or not only mannitol, so paracetamol or whatever it may be. If you take the drug, some headaches or some nausea, vomiting, if you take any leaflet of the medicine, so definitely it will have some adverse events. So that means, so if you take the drug into the humans, healthy volunteers, they may get or they may not get the adverse events. Okay, so those adverse events also mild to moderate to severe. So some persons may have the severe, some persons may not have that, maybe mold, or some persons may not get the adverse event. So whenever the adverse event is coming, so anyhow, whenever you are connected to the clinical nurse, anyhow medical professionals, that means doctors will be available. So subject can free to explain his problem, and the doctors they will attend the problem and they will assess the problem. Suppose if I go and complain that I have the fever, so then they should attend that one. They will check the fever if it is there. So generally they will don't administer the medication. Otherwise it will have the contaminated, that will have the effect our original drug. So that's why most of the cases they don't prefer the medicines. Okay, if really required, they will they will give the medicine for either fever or headache. If 99% they don't give, if really required, so the doctor will administer the co-drug. That means co-drug means, so we are doing experiment for any suppose omeprazole. So during the omeprazole, once you administer the omeprazole, he may get dizziness or nausea something. So a fever or headache, if required, they will give the paracetamol or diclofenac based on the recommended prescription by the doctor. So then once adverse event is documented, if it is adverse event is resolved in that particular period only, no need to follow up. If it is not resolved, then you have to follow up the patient, the subject, that is volunteer, to follow up the adverse event until it resolves. Okay, so concomitant medication, what I said, concomitant medication means, suppose other than our tested drug is called the concomitant medication. So sometimes what will happen, serious adverse events may come. In bioclinical studies, generally serious adverse event will not be. Suppose if any serious adverse event is there, it is also, if it is very severe, then it may go for hospitalization. Okay, so that means whatever the adverse event, headache, a small complaint also from the subject, you should attend. That means that is called subject well-being. So that means until completion of the study and after completion of the study also some period, we should have the monitoring of the adverse events from that particular subjects. So then after that, storage of the samples. So that means sample starting, suppose different points you are collecting the samples, those samples we should sorting to the subject wise for analysis purpose, then we will transfer to the bioanalytical laboratory. So generally transfer of the samples, suppose testing facilities, bioanalytical facilities in the same premises, so no need to directly will transfer within 1 to 3 minutes. Otherwise, suppose testing laboratory is in Hyderabad, then bioanalytical laboratory in other place, then you have to transfer these samples to the bioanalytical facility, then same storage conditions should maintain. So generally we will use the dry ice. Dry ice will give the minus degrees. We will use the dry ice to transfer the samples. So this part is after that, until that part is the clinical phase part. So then come to the bioanalytical part. So already my colleague, that my friend explained, Nageshwar, bioanalytical, how to development the method, how to validate the method, and how to the routine sample analysis. So briefly, that will have the method development and method validation already they will complete. So routine sample analysis, what are the collected samples from the studies? So that is the routine sample analysis. So then development, no need to explain because already explained. These are the different development to sample analysis is the just so brief slide. So like development and the different techniques already he explained, SPE, LLE, and protein precipitation. Then routine sample analysis. So once you analyze the samples, what we'll get? We'll get the concentration each time point. So what is the concentration in the particular sample? We will get that particular concentration from that particular point. So until after completion of the bioanalytical phase, so we should have the concentrations of each time point from each subject. So then, so we have to analyze that data. So to say that these are the bioequivalent or not, both. So that's why that data we have to take the data from bioanalytical laboratory. Then we have to apply. Then we have to go for the calculation phase. So generally, this is the general. Everybody know that one calibration curve. So to know the calibration curve, we will generate from this calibration curve. So we have to calculate our unknown concentration. So extra, this is all Beer-Lambert's law. There is no rocket science in this. So based on this calibration curve, we'll know the unknown concentration in our samples. So these are already slides I'm skipping because of already explained by our friend. So how to analyze the samples. So then come to the bioequivalent. Once concentrations are there, so then other part is pharmacokinetic studies. This is the main important part, because if you get the concentration also, if you don't say if you are unable to say that is both are equivalent or not, then there is no use. So based on some parameters, you have to say that so both drugs are bioequivalent. So that's why. So the pharmacokinetic, generally pharmacokinetic parameters, some parameters are there. So based on that parameters, you have to calculate from the obtained concentrations. So the pharmacokinetic analysis where pharmacokinetic parameters derived from the concentrations are calculated. The concentrations means whatever the amount of the drug present in particular sample we collected. So then you have to apply the statistics to that particular parameters. Then we will derive, we will get some data. Based on that data, we will say that both the drugs are equivalent or not. So statistics means the study of the collection, organization, analysis, interpretation, and presentation of the data. Okay. So then this slide, simple slide. Just we have two drugs. So one is red color, one is like just apple comparison. Just I have to take this slide to understand that one. So can we say these both are equivalent, red and green? No. So like that only, if you want to compare the products, here we are comparing the colors. So this is red color, this is the green color. That's why, so both are not equivalent. So like that, if you compare the brand drug and generic drug, both are equivalent, we should have some parameters. Okay. So these 6 are the generally pharmacokinetic parameters. You know that academic also we come to know these parameters.
సార్ మీ వాయిస్ మళ్ళీ వినపడట్లేదు సార్.
Hello.
Yes sir, now it's okay.
So to compare the both generic and innovator drugs are comparable or not, you will take these parameters. These are the common parameters we will take in bioequivalent study. So all of you know that one, no need to explain, but just briefly, A is means area under the curve, infinitive, from 0 to infinitive. So Cmax means the maximum observed concentration in light over the sampling interval. That means we are collecting from 0 hour to either 24 or 34 hours. So then in that, what is the maximum concentration in that particular sample? That is called the Cmax. And Tmax, you know, where the maximum concentration at what time point the maximum concentration is there, the Tmax. KL, that's the first order elimination rate constant. T off means the apparent terminal elimination half-life of sunlight. Okay. So then if you so general linear trapezoid rule, these all calculations will give the computer only. So no need to do the manual calculation. So generally we will use either Vinodian software or SAS software. So to calculate these parameters. Okay. So the area under the curve, linear trapezoid rules. So whatever is this is if you take one subject only. So that whatever the curve from 0 point to the whatever you collected 24 hours. So it will divide into some trapezoids for calculation. That means so time versus concentration. So what is the time that concentration? They will calculate the entire curve. That means enter from 0 hours to 24 hours, whatever it may be. So entire curve will divide into the trapezoid, then it will calculate the so area and what is the area under the curve for test product and reference product. Both it will be calculated. So then these are the general calculations only. So as I said, so we have the different time points we will collect it. So suppose the example I have taken for up to 10 hours. So then these are the concentrations of particular time point. Suppose 0.25% is 550 something, 5,000 something is there. So at 0.25 hours, we have the maximum concentration. Okay. So those are called the Tmax and Cmax. All of you know that one. So because 0.25 is the Tmax where we get the highest concentration. So that is called the Cmax. So when you see the concentration of the data only, no need to go for any software, we can say the Tmax and Cmax. So this is general typical curve of that particular subjects. So this is the Cmax, and where we get maximum is the Tmax. So what are the area is on total curve is the area under the curve. So then if you know the area under the curve and last concentration, we know C last. That means what are the last time point, what is the concentration, we know. So to calculate the area under the infinitive. So what is the infinitive? Even suppose if you take the drug after 20 hours or 30 hours, or it will divide into half only. So it will continuously infinitive. We cannot that means up to either long, that is 3-4 days, 4-5 days, you cannot collect the samples. So that's why. So based on our last time point, we should estimate theoretically the infinitive point. So that means if you want to calculate the area under the curve until last elimination, that means until last elimination, we don't know exactly. So that's why, so that elimination, that infinity will calculate from the available data from our study. So that means infinite data can be calculated by AUCT anyhow. What is the last time point up to, we can calculate the area under the curve from C last point by KEL. So what is this elimination rate constant? KEL. Okay, so elimination rate constant is the, we need to calculate the slope from the slope of the what are the points obtained. So we can calculate the KEL. If you calculate the elimination rate constant by using the formula, it will be useful to calculate the infinitive. Okay. So then this is the calculation of the elimination rate constant. So what are the curve? These are the time points obtained when you apply the log concentration. So generally we will get some last 4 points or what are the straight line points up. It will software will calculate automatically. So they will give the that software will give the value of the KEL. So if you have the KEL, then automatically. So these are the typical curve. That's what are the curve I have taken with log concentration. So then if you calculate the KL, so we can estimate the AUC infinitive. So this what are the our AUC T, that means T means area under the curve up to collection time points is 9982. Even drug will be remain in the body after last time point also, that we cannot estimate because we cannot continually collect the samples. So theoretically we'll calculate the infinitive. You see, if you see that definitely the AUC infinitive is more than the AUC T. Okay, why? Because we are theoretically calculating until elimination, the complete elimination of the drug from our body based on the available data. Okay, so then half-life, that means T half will calculate from the KEL. That means if you know the AUCT, KEL, we will calculate the AUC infinitive. If you know the KEL, we can calculate the T half also. Okay. That means the time period required for the concentration amount of the drug in the body to reduce the half. What is the meaning of T half? So what is the time taken to reduce the half of the drug from our body? So that is we will call as the T half. Okay. So then even generally your time points also set based on our elimination risk on T half only. So then already we have the data. Okay. That means we have the under the curve, the Cmax, Tmax, everything is, we have the data. So then how do you compare that one? So until that also, we are not saying both drugs are bioequivalent. Okay. So then see, different, generally all regulators having the same criteria. That means 80 to 125%. So statistical means at 90% confidence interval, when you calculate that AUC T and Cmax, so those should be within 80 to, so a ratio, okay, ratio of the test and reference product should be 80 to 125%. If it is within 80 to 125%, then we can strongly say my drug is equivalent to the innovator drug. Otherwise, if it is not within the 85 to 125, 80 to 125, then we cannot say my drug is bioequivalent to the innovator drug. So these are the general, these are the one fundamental criteria if you want to say my drug is bioequivalent to the innovator drug. Okay, generally if you see, suppose when some variable drugs are there, so they will give the relaxation, very drugs which are very variable, maybe 75 to 130, but general 99% the criteria is this one, 80 to 125%. So if you see the simple explanation about the bioequivalent or not, this slide. So suppose our limits are 85 to 125%. So how can we do? Suppose if it is a pink color, if you see, so the whatever the our concentrations are, whatever our calculations with AUC and Cmax both are within the limits, then we can say that our drug is bioequivalent. So then only, suppose if you say this one, so fail to demonstrate because lower level is more. That means our drug is lower than the innovator drug. So then this is failed to demonstrate the bioequivalence. Then here is in between, but some portion of the curve, that the concentrations are, some portion is out of the 125%. So then beyond the 125%, then you can say it is also not equivalent. Here, if you see this is the entirely low side, and this is entirely low side, higher side. So even it is entirely low side or entirely higher side, we can say it is the bio-inequivalent. Okay, so based on this one, you can say the drug is equivalent or bio-inequivalent. So you can have, you should have the doubt. So this side is 80%, why this is 125%? Okay, so this is how the one logic. Suppose we are saying 85 to 125%. If you compare the test to reference ratio, so if you say 80 by 125, how much it will come? But like that, if you go for 125 by 80% also, it should come like that. It is derived. Okay, so T by R ratio or R by T ratio. Okay, so if you calculate 125 80, how much it will come? So 80 by 125, like that. So even though if you compare with test to reference or reference to test, so this should fit into this criteria. So that's why it will come as 80 to 125%. So generally, these are the references. So if these are two differences again, but you can go for each regulatory. Suppose if you search the bioequivalent studies of USFDA, bioequivalent studies of CMAA, so you can get the specific bioequivalence guidelines in that particular respective regulatory from the net. So these are the ICH guideline is the guideline. This is author of the clinical trials. So ICH, you know that one, it is harmonization process, harmonization guidelines. So every country can follow this guideline. So pre-screening revised checklist. So if you go to this checklist, it is available in CDSC website. So in that site, you will get the bioequivalence, everything. It is our Indian site only, CDSC Central Drug Control Organization. So we will get pre-screening revised checklist. In this checklist, what I said, 4 types of or 5 types of applications, you will get the what document required to submit the application. Okay, so these are the brief uh, explanation about the bioequivalent studies. So thank you for your patience and attention. So now you can ask any any your doubts or questions regarding the bioequivalent studies.
Yes, sir. Thank you for giving your precious time, sir, and wonderful presentation. And I hope this may help the participants to connect their theoretical knowledge from Shagul, Brahmankar, which we study in our academics, to the practical sense. Now I request our principal ma'am, Sarada Garu, is principal ma'am is there. Yeah, now
No,
I
ma'am,
request,
she's not.
Now I request Sarada Garu to request
No.
questions from the participants.
Yeah, yes, ma'am.
Very
Thank
short.
you. Yeah. Firstly, I thank sir for giving this such a wonderful information about bioavailability and bioequivalent studies. The most very question we usually have is regarding the generic and the branded drugs and what is the difference. You know, what can we take the generic drugs? This is the most usual questions we will be having, and most of our family members are also uh, come forward. Some may come forward for using generic drugs, and some may obey. Some may not suggest us to go for the generic. So we will be having such doubts, and today we are very clear about these generic and branded drugs. Thank you for your information. Also, again, with the phases you have explained, like clinical, bioanalytical, and biokinetics and statistics. Thank you, sir. Thank you. And here are some questions for you, sir, regarding
Yeah.
this topic. So in the pharmacokinetics and clinical study, we have gone through many calculations like kel calculations,
Yes.
cmax and tmax. So do we have any software regarding these calculations, I mean, to
Most of the companies, they are following Vinolin, madam. Vinolin software.
Inolin software? So
Vinolin, WINN, Vinolin.
okay,
You can search in Google also. Vinolin, you have to purchase the software actually.
okay, so using
So
Vinolin software we can
it is very user friendly software. So what are the
hmm,
concentrations and time points you have to feed that? You can calculate in Excel also. But suppose in
okay,
our organization level, we can calculate in Excel also. All will come in Excel.
okay,
But any statistical calculation very difficult to calculate manually. So that you will, if you have that software, then if you enter that concentrations and time points, automatically you will get the results with curves. What are the curve I
okay,
presented? No? So that
sir.
curve pattern, it will come to the in your hand. That means it will display the curves and everything. It is user
Sir.
friendly software, Vinolin software. So
Okay,
some people may develop the SAS program also. SAS already SAS program they will develop, and so they will go for codes, and they will their own they will. But
okay.
if user friendly is the Vinolin is the user friendly software.
Friendly software. Okay. Thank you, sir. And the other question is in the preparation of protocol for the preparation of sample. So, what are the protocol violations?
Generally, protocol violations are the suppose violations by organizations or violations by violations is generally violating the protocol. So that means
Okay,
whatever you are presenting the protocol. Suppose
sir.
adverse event monitoring is there. Okay.
Hmm, hmm.
Suppose if any subject complain that I have this problem, if you are not attending that problem, it is violation.
Okay.
Okay. And
Okay.
also suppose, so you are saying that so I will collect these points. Suppose up to 24 hours only I am collecting the points. I will collect
Hmm.
the points. So if you beyond that, if you collect the points, that is violation.
Okay.
Whatever it suppose if whatever you are deviating the protocol, whatever you have written pre-written, if you deviate
Sir.
that one, it is a violation.
Okay.
Especially when patients subjects, if you violate, it is a very serious violation. So
Okay.
even subjects volunteers will have rights to complain to the ethics committee.
Sir.
Okay, we have to give the information of that's phone numbers of ethics committee to the volunteers. So because
Okay.
any problem, they will complain to the ethics committee or
Okay.
regulatories. That means regulatory means who has given the permission to conduct the studies. So they will have right to complain to the either ethics committee or that is CDS, that is DCCI. So you have to give the phone numbers of the particular authorities to the patients. That means subjects.
Okay. Uh, okay, sir. And will the age group of the subjects affects the bioequivalent studies?
Definitely, if you take old man and younger man, that means initially I said physiological variation. So
Okay.
female and male physiological variation is different based on the
Sure.
hormones and everything. So like that, if a younger person and older person. So if you
Sure.
suppose older person, generally you have the kidney just function is very less
Weak.
compared to the younger weak. So that
Yeah.
means it will have the elimination effect.
Okay.
Okay,
Okay.
like that. So based on the age groups, definitely physiological variation will be there. If physiological variation is there, automatically our drug dissolution and absorbance will be automatically differ from the groups. So that's why generally bioequivalent studies will take for 18 years to 45 years. Okay,
Okay.
some studies will specific about the age also. So you should not cross beyond 30 age, and BMI should be this age. So that's why it is very particular. Well, while preparing the protocol, only we should select the age group based on the drug and based on the adverse effects. Suppose some drugs may have the serious adverse in older ages. So that's why you
Sure.
should not take that particular population to the our bioequivalent studies.
Okay. Okay, sir. Sir, and there are some drugs which have very narrow therapeutic index. So for those type of drugs, are there any special considerations, while performing bioequivalent
So that's
studies?
why narrow therapeutic drugs, anyhow, if you, that's why. So if you narrow therapeutic drugs based on the development also. So if narrow
Okay.
therapeutic drugs you are not developing properly, so if it release more concentrations and it will have the effect on the patients. So
Patient.
that's why, so not the narrow therapeutic drugs, what are the drug based on their properties, you have to design the study. So
Okay.
narrow, that's why. So, suppose concentration, that means you have the therapeutic index. If it is beyond therapeutic index, what will happen? So it will give the more serious adverse effects.
Oh,
So
okay.
based on that, our formulation should be release the drugs as per that. Suppose it release the more concentration immediately, it will have the
Sure.
more effect. So beyond the window, it will based on automatically it will give us more adverse events.
Sure.
So that's why we have to design based on the drugs. That's why what I'm saying
Drug
when
property.
we are preparing the protocol only, we should consider all these considerations. Type
Okay.
of the molecule and what is elimination rate constant, what is the Cmax, Tmax, everything. So you should consider, and based on that only you have to plan the study.
Okay. Okay, sir. And sir, there are some drugs which are prodrugs. I mean, which are not
Yeah.
active in
Yes.
the formulation state, and when they enter into the body, they converts into active
Yes. Yes.
metabolites. So bioequivalent studies should be conducted to prodrugs or to active and
We
metabolites.
can conduct, no issue, but based on the uh, that's why what are the it will convert and it will give some concentration. It will convert in our body, so it
Mm.
will go to systemic circulation. So
Yes.
whatever I suppose what are the final moieties enter into the systemic circulation, then that
Sure.
will be analyzed in the bioanalytical. Based
Oh,
on that one, our development process will be there. Okay.
okay.
So
Okay.
suppose and one more important is, suppose any endogenous drugs. Suppose if you take testosterones, so already it is on our some portion it is in our body. Okay,
Uh,
so that's
uh,
why you have to collect the pre-sample, whatever you are collecting before drug, before administer the drug. So those concentration you should minus in our main concentration. That's after administration the drug. So that will be balanced. So otherwise concentration will be unbiased. So that's why those type of drugs before pre-drug, how much it is there.
okay.
One more for your question. Suppose when you are taking the participants, what are the zero sample you are collecting before administration of the drug? So
Yes.
that drug should not be more than the 5% of the Cmax. So that
Okay.
if it is more than the, so we should not take that. concentrations into the calculations. So it
Okay.
should be less than the 5%. So
Okay.
then only that subject you have to take the calculations. If that means suppose before day. So that's why. So one day before, 24 hours, 12 hours before only, you have to participants should come into our center.
Sir.
That means total 12 hours you should not give anything. So that whatever the drugs he has taken prior to without information, that shall be eliminated.
Okay.
So then only we will administer the drug after general 10 hours, 12 hours only we will administer the drug. So once he come to the center, he will stay there. Then only we will administer the drug. Otherwise, what happen? Immediately if he come, if administer means prior to that only, if he take the drug, they don't say
Uh,
us. Some people they don't say
yes.
us. So
Yes.
suppose I am giving the some drug, same drug we have taken some other purpose. So when you give immediately the drug, that immediately that double dose will. That way concentration will be more, some serious adverse will come to the subjects. So that's why 12 hours before only we recruit the subject. They should stay our center. So then before the drug, again we will monitor the vitals. Then we will administer the drug. Suppose even though if elimination is more, then they may not eliminate the drug from our body completely. So that's why some criteria is it should be less than the 5% of the Cmax. Then only that person uh, time points are that person will enter into the calculations. Otherwise, you have to remove that person into the that particular data into the calculations.
Calculations. Okay.
Yes, that is one of the thumb rule.
Okay. Okay, sir. Sir, can we have any example, like any protocol format you have done?
Protocol format you can search in Google, madam.
Okay. Okay,
Most
sir.
of that, if I have, I will forward to you.
Yeah, yeah. Okay, sir.
Okay, but actually, no researcher will go for bioequivalent studies. So
Okay.
if they go, that is good, but because it is very expensive.
Mm,
So because
yes. Okay.
recruiting and subjects, you have to pay money. That is very bioanalytical, also very expensive. So
Sir.
very rare compounds we go for HPLC. Most of the drugs most of the analysis for bioanalytical. That means LCMS only.
Yes.
Because
Yes.
very picograms or nanograms is there. So then majority will go for LCMS.
Okay. Okay, sir. Thank you. Thank you for solving these questions.
And thank you,
And
ma'am.
over to.
Yeah, thank you so much, ma'am, and thank you, sir. And you exposed us for about drug product performance, starting from the basic, like purpose of bioavailability studies, relative and absolute availability. Not only that, covered the design, study designs. Actually, we are in need of all these things in our inform course also. And protocol, ma'am asked about the protocol because we have that protocol preparation for the conduct of bioavailability, bioequivalent studies according to guidelines. Just they asked us to study this protocol, how to prepare the protocol to conduct this in conducting the BA studies.
So protocol preparation, we can, that's why, ma'am, protocol preparation, whatever shown the parts of the protocol is the
Yeah.
protocol
So
preparation.
the same thing.
But if you want to conduct the study, you should take the approval from the ethics committee.
Yes,
That
yes,
approval is mandatory. So you
yes, sir,
simply
yes, sir.
preparing protocol, you cannot conduct the study. That is
Yes.
unethical.
So
Okay.
we need only the simple preparation only, sir, just for our
Preparation of the
practical
protocol.
purpose. Yes, sir.
Yes.
So okay, madam. If I have, I will for any
Sure,
protocol,
sir.
previous
In that
protocol, I will
connection,
share to you.
yeah, in that connection, if we have any doubts
So
regarding this,
actually,
we will let you.
even that is suppose. So generally, if you give one molecule, also you can search the literature. You are able to. That's why if anybody is industry experience, they can prepare the protocol their own way, because the time points, as I said, what are the how to set the time points? It is one. Setting the time points also, you should have some data. That means Tmax and elimination concentrations. So generally, we should keep the time points at least 3 half-lives. So as I said, Tmax, half of the drug should be eliminated within 2 hours. That means Tmax is 2 hours. So time points, you should get at least 6 hours. Why? Because by 2 hours, it will 50% will be eliminated. By 4 hours, it will eliminated 87%. By 6 hours, it will eliminated around 97%. That means half-life of the drugs, based on the half-life of the drug, our time points will be depend. This is the thumb rule. So anyhow, I will send one protocol if anything is there, just you for your reference.
Sure, sir. Like if you have any chance, like come to this, please come to our college. So that we'll be having offline session also. Thank you
Sure.
very much, sir.
Sure.
Thank you, and dear participants, thank you very much. And you please fill the Google form, whatever you are going to have in your WhatsApp group. Thank you, Madam. Sir, we can leave, I think.
So thank you, madam. Thank you for giving this opportunity. So feel free to ask me if anything, either quality control related or if anyhow, because generally what we when we were studying, we don't have this facilities or this.
Yes, sir. Yes, sir.
So
Yes,
okay,
sir, that's why.
okay. Even so, that's why if you have any quality control related or QA/QC related, okay. So then if you have any this type of sessions, feel free to so ask
Sure,
me.
sir.
I will participate whatever extent is possible. Okay.
Even I thank very much, sir, and at the same time, I thank Shivarati, sir, to introduce you to our college.
Yeah, Shiva and me are both are 8 years roommates now.
Yes,
M Farm and B Farm.
actually, he also suggested your name and at the same time. So thank you very much, sir, for your
Okay.
presentation.
Right.
Thank you, sir.
Thank you.
Ma'am, can I end the meeting?
Sure.
Okay.